All right, thank you very much for having me. I will be talking about the role of vascular aging in cognitive decline and Alzheimer's disease. We saw some slides on major risk factors for Alzheimer's disease, and among those was age itself. And some people have argued that age is potentially the most important risk factor for the sporadic form of Alzheimer's disease, perhaps in addition to the apolipoprotein E4 allele. But what is age? It's this chronological kind of measure. It's not in itself a biological measure. It's sort of a proxy for a bunch of biological processes. So I'm going to talk about one of those, which is the vascular aging. So I was interested for a long time, basically since the beginning of my career, in the connection between the brain and the vascular system. It's not at all clear how these two are related, but we know that cardiovascular risk factors, for example, are risk factors for Alzheimer's disease. And basically every vascular risk factor that's been studied seems to be related to the disease. But again, we still don't really understand what the mechanism is, and it's not clear whether this is something that's happening in the blood vessels in the brain, the cerebrovasculature, or whether it could be a more distant link to actual diseases occurring in the heart or the beginning of the aorta. And so as a graduate student, I studied aortic atherosclerosis in animals. And so naively, as I entered into the Alzheimer's disease field, I was interested in this question of the vascular connection with Alzheimer's disease. And I started to do some literature review on aortic diseases. And I actually found a very interesting and rich literature on the relationship between vascular aging and arterial stiffness. So I learned that the large arteries in the body, such as the aorta, biggest blood vessel coming off of the heart, are very stretchy. They're among the stretchiest tissues in your body. And so part of the aging process, which is actually due to a convergence of many different biological processes, these elastic fibers that convey that stretchiness to those large vessels, it's called elastin, starts to break down and degrade as we get older. And this is something that happens to most everybody. It's associated with vascular risk factors like high blood pressure, but it can happen even in the absence of those clinical risk factors being detectable. Those elastic fibers are then replaced by collagen, which are stiffer fibers. They don't really stretch. And so the end result is that you get a hardening of these large vessels. And interestingly, one of the purposes of the stretchiness to these vessels is that they actually buffer fluctuations in pressure as a pulse wave is ejected from your heart and goes throughout your arterial tree to feed all your different tissues. And so the body's sort of trying to solve a problem. How do we need to have steady blood flow to tissues that need continuous nutrients, oxygen, but yet the heart is a pump that works in an oscillatory way as it's ejecting the pulse wave. And so the solution to this problem is what's called the Windkessel effect. So this is where the large arteries like the aorta will stretch and recoil in response to the pulse wave, and that ends up buffering this fluctuation in flow and pressure at the more distal, smaller arteries and arterioles, and ultimately you get steady blood flow in the capillary beds that perfuse major organs like your brain, that require torrential amounts of blood flow. So they're very demanding of blood, and that it needs to be continuous. And so as these large arteries begin to stiffen and harden, you start to kind of lose this Windkessel effect, and you end up with more, the fluctuation in pressure gets pushed out to those distal tissues and is thought to be harmful. So for example, the increased stiffness of these large arteries is associated with things like kidney disease, because the kidneys also need to be perfused at very high volumes of blood, and they're getting the consequences of this fluctuation or pulsatile pressure and blood flow to the tissue. Another consequence is that the pulse wave moves quicker. And this is just like a physics thing where fluids move faster through a stiff tube than a stretchy tube. And it's the same basic idea. It's not buffering fluctuations in pressure. So how do we measure this? Well, it turns out there's a very simple measure for arterial stiffening, and this is brachial artery pulse pressure. This is like a regular blood pressure measure you get when you go to the doctor. You get systolic pressure and diastolic pressure. Usually we look at them separately, but if you look at the difference between systolic and diastolic pressure, you get this pulse pressure index. And as it turns out, pulse pressure really goes up as we age. When you look at the individual indices of blood pressure, systolic pressure goes up, but mean arterial pressure doesn't really increase that much with age, and diastolic pressure actually starts to drop off. So you get this widening of pulse pressure. And this is thought to be due, again, to this change in the way the pulse wave is propagated throughout your arterial tree with vascular stiffening with age. So why does this matter? Well, it turns out this probably plays a role in the fact that older adults end up with a lot of what we call isolated systolic hypertension. So basically, people get high blood pressure. And a lot of the antihypertensive drugs that we use don't really address this pulsatile component of blood pressure. They may lower blood pressure by changing the total blood volume or through some other mechanism. And so only certain medications are really targeting this stiffness problem. They're called de-stiffening medications. But there's a lot more work that would need to be done to address this issue. So how is this relevant to cognitive decline? Well, it turns out, way back in 2003, there was a paper that came out showing that people over 75 years of age, pulse pressure was basically a risk factor for Alzheimer's disease, similar to many other vascular risk factors. And then Sherry Waldstein did an analysis as part of the Baltimore Aging Study, a large community-based cohort, and found that increased pulse wave velocity, again indexing arterial stiffness, was associated with decline in memory ability on a word list learning task, very sensitive test of memory in this population. And this effect of pulse wave velocity on memory seemed to get greater with age. So the older people got, the larger their pulse wave velocity, stiffer the arteries, and the worse their memory performance was. And then as a postdoctoral fellow, I investigated pulse pressure in relation to a lot of different cognitive markers that we tend to see changing early in the Alzheimer's disease process, and found that it was associated with other things, including executive abilities and other language-based tasks that show early decline in Alzheimer's. Gary Mitchell is another person who's done a lot of work in this area and has actually developed many other indices to look at stiffening not only in the aorta, but up into the carotid arteries and at specific branch points to try and figure out if stiffening more proximal to the brain may be associated, again, with cognitive decline, but also measures like brain volume, which we know people start to show some brain atrophy early on in the Alzheimer's process, and also subclinical or occult cerebrovascular lesions that show up on people's MRIs even if they haven't had a clinical stroke. And sure enough, he found that a number of his indices correlated very highly with these cognitive measures and brain measures in a large Icelandic cohort. But in trying to get a better grasp on this, I was really interested in understanding whether pulse pressure is just causing this kind of subclinical vascular disease that's adding to your cognitive burden in people who also have Alzheimer's, or whether it has a direct link to the pathophysiology of Alzheimer's disease. And so one way that I started to look at this was to investigate pulse pressure in carriers of the ApoE4 allele to see if there was an interactive effect. And in a large cohort from the Framingham Heart Study, one of the longest running studies of heart disease in the world, we found that pulse pressure in midlife, people in their 50s, predicted the future decline some 7 years later on cognitive tests, and also showed relationships with medial temporal lobe atrophy or hippocampal atrophy that we tend to see in the prodromal period of Alzheimer's disease. And these associations were greater in people who were carriers of the ApoE4 allele, which suggests potentially this genetic interaction. And this was interesting because a lot of basic science has been done to connect ApoE4 with vascular dysfunction in the brain, although the mechanisms are still getting worked out. And so I started to think that maybe there is a connection directly with Alzheimer's disease, and wanted to investigate this with some actual pathological measures of Alzheimer's disease itself beyond cognitive changes. And so we had an autopsy series of over 100 brains of people who had gone on to develop Alzheimer's disease and had been given a definitive diagnosis at neuropathology, which is the only way we ever know for sure whether somebody had Alzheimer's disease. And they had had their blood pressures taken, naturally, as a part of the study many years prior to their deaths and autopsies. And so we looked to see if antemortem pulse pressure predicted the presence of different pathologies in these Alzheimer's patient brains many years later. And sure enough, we found that pulse pressure was related to vascular disease in these Alzheimer's patient brains. We actually see quite a bit of vascular disease in Alzheimer's patient brains, even though it's not necessarily part of the diagnostic criteria. And so pulse pressure seemed to index that. So for every 5 millimeters of mercury increase in pulse pressure, there was a 36% increase in the odds that people would have vascular disease in their brains rather than having the sort of pure Alzheimer's disease. We also found a linear relationship between the extent of pulse pressure elevation and the amount of cerebrovascular disease people were exhibiting. But we didn't find a relationship with our markers of Alzheimer's pathology at autopsy. And so I started thinking, well, this is just related to vascular disease. Again, it's probably not linked to the underlying Alzheimer's process, which we define as beta amyloid plaques and neurofibrillary tangles comprised of phosphorylated tau protein. We didn't find those relationships. But there are lots of caveats to neuropathological studies. We can't control when it is that someone's going to pass away, and so we cannot measure our risk factors in line at the same time as the pathological factors, and we have to wait until someone has passed away, at which point, in the case of definitive Alzheimer's disease, they tend to have a lot of disease burden at that point. And so it's hard to find meaningful relationships. And so I was, with the advent of cerebral spinal fluid based and other biological markers that we just heard about, such as beta amyloid protein buildup in the brain, we can now measure this in people, and we call this preclinical Alzheimer's disease. We were actually able to start to investigate links between arterial stiffening and the Alzheimer's process much earlier on, at the earliest stages of disease, some potentially up to 25 years prior to people even being diagnosed with dementia. So we still weren't convinced that arterial stiffening may not have a role. But one question was, how would this be related to Alzheimer's disease? It's clear that arterial stiffening would maybe convey risk for stroke or cerebrovascular lesions, but how would it connect to Alzheimer's disease? And so in looking through the literature, we found that some people, some neuropathologists, had hypothesized that perhaps arterial pulsation plays a role in clearing beta amyloid from the brain. We know that patients with Alzheimer's disease have a hard time clearing amyloid beta, and it's not necessarily that they're producing too much of it, but that there's something wrong with the ability to clear it out, at least in the sporadic form of the disease. And the brain actually we're still not perfectly clear how the brain clears toxic waste products, metabolic waste products. It's really a basic question that we just don't know in neuroscience. And the rest of the body has a lymphatic system and a musculoskeletal pump, where when you're moving, you're pumping lymph through a series of one-way valves, and this helps clear out toxins from your muscles and other tissues. But the brain doesn't have muscles and that sort of thing. And so you've got this kind of fixed cavity, and it's thought that arterial pulsation may play a role in kind of pumping out toxins and metabolic waste products from the brain. But actually, a lot of breakthroughs have been coming through very recently in this basic field. And we now know that there's this thing called the glymphatic pathway, which is a new mechanism that's been discovered where a convective bulk flow mechanism actually sweeps toxic metabolic waste products and other proteins such as amyloid beta out of the interstitial fluid, the space between cells. And then these run along these perivascular channels, little tiny spaces around blood vessels. And these little spaces are so small that that's part of the reason why it's been so difficult to study. But this was a major breakthrough. And it's been found that this pathway is turned on during sleep and probably is a big part of why we sleep and is actually a potential connection, again, between sleeping and Alzheimer's disease risk. And even more recently, there have been another major neuroanatomical discovery. So one criticism of this glymphatic pathway mechanism is that it's sort of a closed loop. The fluid goes from the arteries through the tissue to the venous side, but then it's just back in the cerebral spinal fluid. So it wasn't really clear how it's getting out of the brain and down into the lymphatic system. But very recently, a paper was published in Nature showing that the brain actually has lymphatic vessels, whereas before we didn't think that it had those vessels. And so that provided a more clear kind of picture that these toxic proteins are swept out of the brain along these perivascular spaces, and then they connect up with the lymphatic system actually within the brain. And Micah Nedergaard, who has characterized this pathway, actually has shown that in animals, the small arteries in the brain, the arterioles, actually will show less pulsation. They seem to stiffen with age, and this can interfere with the clearance of amyloid beta from the brain. So this provided a mechanism whereby arterial stiffening may actually play a role in the clearance of amyloid beta and thus Alzheimer's disease pathophysiology. And sure enough, the first study that I'm aware of came out of Banner Arizona Health, Langbaum, and from Eric Ryman's group, showing that brachial artery pulse pressure was associated with increased amyloid beta deposition in the brain using PET scans. And then since that time, a number of other groups have replicated this finding, and we found that other markers of arterial stiffness are associated with amyloid beta deposition in the brain. But one question that I had early on was, okay, it's related to amyloid beta, but what about the neurofibrillary tangles, phosphorylated tau protein? That's the other side of Alzheimer's disease, and we know that it's important because it correlates well with clinical symptoms of the disease. And when we just measure the amyloid beta deposition with our PET scans, it doesn't tell us anything about what's happening to the tau protein. So we did a study in cognitively normal older adults that were very healthy. They actually had to have a low BMI, below 30, and body mass index that was lower. And they couldn't have diabetes, and they also couldn't have multiple heart attacks. And so we were getting a sample that was relatively vascularly healthy. And this was important because we were wanting to know, does arterial stiffening, which we know is independent potentially of vascular risk factors, still link up with Alzheimer's disease early on, even if you are healthy in terms of what we normally think of as clinical risk factors for vascular disease? And so we investigated the relationship between brachial artery pulse pressure and our cerebral spinal fluid biomarkers of preclinical Alzheimer's disease, so amyloid beta protein and p-tau, or phosphorylated tau protein. And sure enough, we found that pulse pressure was associated with lower levels of amyloid beta in the spinal fluid, which is thought to be indicative of retention of amyloid beta in the brain. So in other words, this is a marker of preclinical Alzheimer's disease. We also found that it was associated with increased levels of p-tau in the spinal fluid. And this is the other component of preclinical Alzheimer's disease that comes from our CSF biomarkers. And when we looked at the ratio, it was even more significant. So this was striking because it was the first time where, in my group's lab, we were able to actually find a connection or a link, an association between arterial stiffening and the pathophysiological markers of Alzheimer's disease. And in fact, pulse pressure is one of the few readily available indicators that is linked to preclinical Alzheimer's disease. But one thing we noticed that was a bit curious is that the relationship with amyloid beta wasn't as salient as the relationship with phosphorylated tau protein. And this was somewhat surprising because we had thought the mechanism would be impaired clearance of amyloid beta. So this suggested that in studies where we're linking this up with amyloid beta but not measuring tau protein, maybe the relationship is really with tau. And so this has bearing on our preclinical Alzheimer's disease staging schemes. And so the general idea, as has been described by Clifford Jack and other people, is that preclinical Alzheimer's disease goes through a series of stages. So stage 0 is all the CSF biomarkers are negative. You don't have amyloid beta retention, you don't have phosphorylated tau elevation in the spinal fluid, and you're cognitively normal. Stage 1 is you get some amyloid beta buildup in the brain, but everything else is normal. Stage 2, you have amyloid beta buildup and now showing p-tau buildup, which is thought to indicate neurodegeneration. And then stage 3, you have the biomarkers of amyloid beta and p-tau indicating preclinical Alzheimer's disease, and you're showing some subtle cognitive decline. But this is very early on, prior to people even experiencing mild cognitive impairment, which is a later, or thought to be a later prodromal stage of the disease. And so I wanted to know, is arterial stiffening really related to amyloid beta itself or p-tau itself, or is it related to the combination? Thinking that this could give us a clue as to the mechanism by which arterial stiffening is impacting Alzheimer's disease. So in a large cohort from the Alzheimer's Disease Neuroimaging Initiative study, we had many individuals, over 870, who had cerebral spinal fluid biomarkers. And we divided them into groups based on their biomarker profiles. So we had the stage 0 group, which is the white bar. They had no biomarkers elevated. We had a yellow bar group, that's just amyloid beta, thought to be stage 1 of the disease by the Sperling criteria. And then the orange bar, which was only p-tau elevation. Now in the staging scheme, just having p-tau elevation doesn't really fit, and that group has been termed the SNAP group, or suspected non-Alzheimer's disease pathology, but it turns out a substantial number of people actually show this profile, and they don't really seem to differ in ways you might suspect if they had another disease. So the profile staging scheme has been modified somewhat, and we now recognize that some people may show p-tau elevation before they show amyloid elevation or something else. And interestingly, we found that in the young old adults, age 55 to 79, they were showing elevated pulse pressure if they had high p-tau levels. But they didn't show any elevation in pulse pressure if they were just amyloid positive. And then when they had both biomarkers positive, that's the red bar, there wasn't really a change. So really it was associated with the p-tau elevation. The only way to really disentangle that was to have a large sample where we could look at people who were just amyloid positive or just p-tau positive. But interestingly, in the very old, people who had the highest pulse pressure, right, because we're indicating vascular aging, most advanced vascular age, highest pulse pressure levels, also highest risk of dementia, they showed more of a dose-dependent sort of an effect, where stage 1 group doesn't really show much elevation, maybe a little bit higher. The SNAP or p-tau elevation group a little bit higher than that, and then the people who had both biomarkers positive were clearly showing much higher pulse pressure levels than anybody else in the study. And so how can we account for this? I have a model that I think is relevant. We know that arterial stiffening begins in the aortic arch. So in these young old people, they probably don't have cerebrovascular stiffening in the brain. That's something that happens later. Arterial stiffening tends to progress from the aortic arch to more distal tissues. And so early on, I would hypothesize that we have a hemodynamic mechanism where the blood pressure and blood flow changes that I described earlier are likely potentially resulting in neurodegenerative change through hemodynamic injury to the brain. Whereas in the very old group, we may actually have stiffening of the brain vessels themselves, which is actually very difficult to measure. We don't have a lot of great measures for that process. And if we have stiffening of those vessels, then perhaps we start to get difficulty with clearing amyloid beta along the perivascular channels that I described earlier. In a reanalysis of this same data, but with the whole group, not splitting between very old and young old, I applied the more standard Sperling criteria, and again, found the same thing, that stage 1, pure amyloid, doesn't really elevate. The SNAP group shows elevation in pulse pressure. Stage 2 is sort of similar. Stage 3, we have individuals who are showing not only amyloid and p-tau, but also subtle cognitive decline. Those individuals were showing even greater pulse pressure levels. And then if you just count the biomarkers, irrespective of their order of appearance, again, you get a similar kind of dose-dependent effect. And keep in mind, this is a group of people who are cognitively normal, so still a very healthy early stage of the disease process. So I mentioned that we don't have good measures of cerebrovascular stiffening, but we're trying. So as a postdoctoral fellow, I decided that I wanted to try and index vascular stiffening with what we had available. And I figured that stiffer vessels show greater vascular resistance, and we know that. And so we can calculate cerebrovascular resistance as the difference between arterial pressure and intracranial pressure divided by cerebral blood flow. The idea is if you increase pressure, you should increase flow, unless there's some kind of resistance happening there. And so this is the general model that we used to try to index stiffening in the brain using arterial spin labeling MRI. This is an MRI method that we use to measure cerebral blood flow. So blood is magnetically tagged before it enters the brain, and then with the MRI machine, we acquire tagged images and non-tagged images and then subtract them. So we get something that's proportional to cerebral blood flow. So we had blood pressure measures that we were taking prior to people scanning, and we purchased this device that measures blood pressure while people are in the scanner, which is something that's not commonly done. And we were able to then measure the ratio of blood pressure to cerebral blood flow during the scan. So the idea is, again, if you have high pressure but low flow, then perhaps you have some increased vascular resistance in your brain, vascular stiffening. If you have low pressure yet you have high flow, it suggests low resistance. And if, in fact, if you regress or correlate blood pressure with cerebral blood flow in patient brains, you have various areas that seem to show an association between pressure and flow. And when we compared cognitively normal individuals to those with Alzheimer's disease on this measure, we found that there was increased cerebrovascular resistance index, what we call this measure, in various brain areas, including the medial temporal lobe or the hippocampus, where we know Alzheimer's disease tends to strike early on, and also an area called the precuneus, which we know shows metabolic changes and cerebral blood flow changes early in the disease. And a similar pattern was observed when we compared mild cognitive impairment individuals with Alzheimer's patients. We also found inverse relationships. So higher cerebrovascular resistance was related to reduced volume of the hippocampus when we measured vascular resistance in that area. We also found that high vascular resistance in other parts of the brain, like the caudate nucleus deep in the brain, that positively correlated with vascular lesions in people's brains. And then finally, we found an inverse relationship between vascular resistance and cognitive functioning in patients with mild cognitive impairment and Alzheimer's disease. So we have reason to believe that this is linked to Alzheimer's disease, cognitive functioning, and also vascular lesions. We also found that aging itself seemed to be associated with increased cerebrovascular resistance in a variety of brain areas, and that this involved more areas than we would see if we just looked at cerebral blood flow, irrespective of someone's blood pressure level. So this is continuing to be investigated, and we're trying to refine our MRI-based methods to try and better index vascular stiffening in the brain. And there was a big breakthrough very recently, a few weeks ago, where Danny J. Wang from UCLA published a method whereby he could measure intracranial vascular compliance, or the stretchiness of blood vessels in the brain. And so it's sort of similar to what I was indexing with vascular resistance, except he's using a newer method of arterial spin labeling that allows us to actually look at changes in the blood volume within vessels during changes in blood pressure. And so as blood pressure oscillates, you should see a pulsation of the vessels in the brain if they're not stiff. But now we're able to look at the ratio of these blood pressure changes to changes in vessel diameter and show that actually with aging, we have less stretchiness of our blood vessels, less vascular compliance. And so we're very interested in applying this same method to, again, preclinical Alzheimer's disease to see if not only stiffening in the heart, but as it progresses to the cerebral vasculature, is this playing a role in Alzheimer's disease? And this matters because it would kind of open a new target for potential therapies, de-stiffening medications, and also help us to just better understand how the vascular process of aging can actually impact Alzheimer's disease. And to further refine our hypothesis, we've also got, in collaboration with Berislav Zlokovic, an investigator at USC, a panel of markers of the neurovascular unit functioning. This is a critical organ that integrates the vascular system with the brain, actual neurons. It regulates blood flow to the brain and also what we call the blood-brain barrier, which is responsible for stopping toxic blood products, metal ions, proteins, and other things that would cause brain damage if they got into the brain. So we can measure indicators of neurovascular unit function in the spinal fluid. And one hypothesis that we have is, again, that this injury to the brain through changes, oscillation in cerebral blood flow and blood pressure related to arterial stiffening, even in the arch early on, may cause dysfunction of the blood-brain barrier and the neurovascular unit. And potentially that could be why we're seeing a link with neurodegeneration in the preclinical biomarkers, irrespective of the amyloid beta changes that we see. We also know, again, that it progresses from the aortic arch up into the brain, and so we're going to apply our new measures of cerebrovascular stiffening in the same study to see if those are related to, again, amyloid beta protein buildup due to an impaired clearance of amyloid beta from the brain. So I'd like to acknowledge my collaborators at USC, Helena Chu, the director of our ADRC, Berislav Zlokovic, who I mentioned, also my collaborators at UCSD, where I did my postdoctoral work, so Mark Bondi and Thomas Liu, and others at the ADRC there, Douglas Galasco. And I'd also like to acknowledge participants in all of the studies that I showed data from, so the Alzheimer's Disease Neuroimaging Initiative study, the Framingham Heart study, and other local studies. So I'm happy to take questions at this point.
I'm going to take the first one, because I'd just like a restatement, if you will, of the post-pressure measurement and how that influences the lymphatics and the taking out the garbage hypothesis.
Okay. So the idea is that we have to clear toxic proteins from the brain, such as amyloid beta. And it's not perfectly clear how this works in the brain, but we know some things. And it's thought to be the case that pulsation of the vascular system in the brain, the cerebrovasculature, facilitates the removal of toxic proteins and metabolic waste products from the brain based on, again, animal studies. And so if stiffening is happening, those vessels in the brain, then they're not going to pulse as well. And so that lack of pulsation could stagnate, if you will, the clearance of these toxic proteins from the brain, and could explain why stiffening, markers of stiffening in general, do seem to relate to amyloid beta levels in the brain, particularly in people who are over the age of 80, where we might expect them to show some vascular stiffening in the brain. I had this person.
Over that way, and then in the back. 1, 2, 3. Okay.
Who's first?
Oh, go ahead, right behind you. Do one.
Will you comment on sleeping position as to pulsation and clearing out the sludge?
Yeah, I know that this has been recently studied and that the lymphatic pathway is facilitated differently based on your head position while you're sleeping. And I believe it's with people laying on their sides that facilitates, if your head is laying to its side, that facilitates the clearance from the brain. But I'm not the expert on that particular area. It's a new finding, very interesting.
Okay, over there.
Okay, I want to know if you had a heart bypass, is there a connection if you had vascular dementia?
So it sort of depends. Cardiovascular disease is a risk factor for dementia, but that doesn't mean that just because you have cardiovascular disease or had a bypass graft or something that you're going to have dementia. It's just associated with increased risk to some degree. And we're not talking about the kind of risk you would get with, say, the apolipoprotein E4 allele, but a small increase in risk in some studies. And it's not perfectly clear what the connection is between coronary artery disease and Alzheimer's disease at this point.
Okay, in the back there.
This is going back to the amyloid beta going through the lymphatic system. Does that mean, does it go through the urine eventually, or I mean, how does it leave the body? By way of urine or what?
I actually don't know the answer to that question, exactly how it's excreted once it leaves your brain. There are multiple ways that amyloid can be removed from the brain in addition to this clearance mechanism I'm talking about. It can be enzymatically degraded by a series of different enzymes, just broken down. It can also be cleared across the blood-brain barrier, just pumped directly out of the brain into the bloodstream. Hopefully that answers your question. I
Well-
don't know if it's excreted in urine or not. Maybe I'm certain somebody else knows the answer to that question.
Yeah,
here.
well, I suppose one could examine the urine and find, and look for amyloid beta, or maybe that's not possible. And the other question is this: the brain creates this amyloid beta. Why does it do so to begin with? What is the function?
Yeah, not being an amyloidologist, I will try to answer that question. My understanding is that, yeah, I'm certain there's more than one in the room. But my understanding is that we still don't really know what the homeostatic function is of amyloid. But there are various forms of amyloid, some of which are more toxic than others.
Right here. In the clinical trials, do they do this testing of this difference in the two different kinds of blood pressure?
Well, certainly blood pressure is likely to be collected in participants in clinical trials, but yeah, and so that's the nice thing about pulse pressure is it's probably been collected in almost every study of Alzheimer's disease, so it's very easy to measure, and is also clinically available to almost anybody who ever sees a doctor or a nurse. So yeah, it's available to be investigated.
Another question. You mentioned de-stiffening medications. Do they exist yet?
Yes, so there's classes of antihypertensive medications or blood pressure lowering medications that have de-stiffening properties. And the one that's probably most notable for this are the AT1 receptor or angiotensin II AT1 receptor blockers, also called ARBs.
Okay, in the back, and then we'll come over here, sorry.
Here's a question.
Thank you.
I understand that diabetes is one of the risk factors for Alzheimer's disease. And I wonder if anybody compared the brain of the Alzheimer patient, diabetes with the normal brain, having higher chance of accumulating amyloid, beta amyloid, or tau protein. So
Yeah, so the question is whether people with diabetes, or whether diabetes has been linked to preclinical biological markers of Alzheimer's disease. I believe that there have been attempts to establish that link, and I think that there have been results in both directions. So some people finding an association, others don't, so I think it needs to be studied further, and that there's a very interesting and important connection between diabetes and Alzheimer's disease through a lot of complicated potential mechanisms, and there's a lot of people studying this now, including Suzanne Kraft, who actually has a clinical trial of intranasal insulin administration.
I was interested in some of your last comments about the blood-brain barrier and its increasing in efficiency. Does that have some other implications for the brain diseases, et cetera, and do we know?
Yes, so the blood-brain barrier opening is definitely thought to be a bad thing. And some of the early studies coming from Dr. Zlokovic's group have shown that people with mild cognitive impairment show increased opening of the blood-brain barrier. And other studies have suggested that it could be linked to other neurodegenerative diseases. So it's an active area of interest and research.
You mentioned the differences between the two blood pressures as being an indicator. Are there normal ranges that are acceptable, and is there ranges between the two pressures that signal a problem?
Yeah, this is a fair question, and a real limitation of blood pressure is that it's highly variable. So The simple pulse pressure measure is nice because it's available all over, but people's pressures go up and down, and they vary quite a bit. So it's hard to establish clear cutoffs. We have cutoffs that are used clinically for diagnosing hypertension, but pulse pressure cutoffs are more complicated because you have two different measures. So pulse pressure in someone with really high diastolic pressure might mean something different than high pulse pressure in someone with really low diastolic pressure, for example. And so that's been difficult to establish those kinds of cutoffs. Conventional cutoffs have been applied in some studies, but I'd hesitate to put my money on a specific cut point. It probably depends, again, on the different components of pulse pressure and the age of the person, and perhaps other factors affecting blood pressure and risk.